Okay, so I'm going to build on some of the things that I've introduced before. So I hope that tutorial will help a little bit to understand more what's happening here. And I'm going to start with one of these networks where I'm only showing you the connections between the particles. So you can see the disorder. I hope you can see the heterogeneity. And what is happening here is that we are sharing the system this way. And at the same time, in this visualization, we are looking at local tension. And whenever you see something become thicker is because tension is accumulating there. So if I start from the beginning, this is going well into the nonlinear regime where the gel is starting to break. That's when things become blue. But you can see that even here, which is in the linear regime, there is actually a subset of the structure in which the tension accumulates. And so this builds on the idea that actually in this type of network, part of the question of trying to understand what is the disorder and what is the organization and how they are connected with each other to produce some functionality, it's about maybe identifying which subset of the structure it's really important for the mechanical response. And is there a subset of the structure that does that? If this is the case, just looking at the structure is not it's not possible to explain the rheological behavior that you see. And so this is a story about that. So I go back to my little picture of the gels and the type of structure that I have in mind. And I want to bring that back because the other question that this raises is that, okay, they all look very different. These are systems that have nothing to do with each other. I mentioned connection with biopolymer networks, with other supramolecular associating systems in all different contexts. And so the question is, do this, one of the difficulty in describing and predicting the mechanics and the rheology of these materials is that precisely the structures, they all look just very different. And so is there actually some organizing principle that maybe correspond to this subset of the structure that actually controls most of the mechanics? Or it's just that I should just simply forget about this and then do a theory for each single structure and just care about describing every single structure separately. So these are structures that we've obtained from the model that I was describing before, right? From the very far left, this is obtained at a given volume fraction, relatively small, with a given protocol that here I characterize with this parameter that is what we call the gelation rate, which is a way to do the gelation in the simulation. Then if I go to the center, here I'm actually just increasing the volume fraction a little bit. Certainly the structure changes, becomes a little bit denser, but how different is different, it's not that obvious. And then on the right, between the center and the right, I actually have precisely the same volume fraction, but I've changed the gelation rate. And so the structure also changes a little bit. Again, some changes that, doesn't seem, it's again messy and it's not clear if there is really some very important difference. But if I look at the rheology of this, so these are the results of oscillatory viscoelastic tests in simulation that we've done precisely with the chirps technique that I was mentioning before. And so you get the spectrum over a broad range of frequency, these spectra, the G prime and G double prime, the storage modulus and the loss modulus, they change over broad range of frequency, which is what you see in the real materials. But also the structure changes a little bit in the way I described, and the moduli is all over the place. It changes by orders of magnitude, not only the moduli, but also the range of frequency over which the moduli changes more significantly. So there are clearly, even if I see small changes in the structure, there are dramatic changes in the rheology, which then triggered the type of question that I was asking before. So I can do, my little network representation, and here maybe I can see a bit more that there are changes in these networks. But again, to characterize how these networks are different, it's kind of it's kind of difficult, and I'm left with the fact that, okay, this is just that I have to do a separate description for each of these gels. So if I take now, because we can do these simulations with very large samples and compute very quickly the viscoelastic response of a broad range of frequency, then you can do, these are like, I don't know, maybe 11 or 13 gels where we vary both volume fraction and range of and gelation rate. And here what I'm showing is a representation of the viscoelastic response, that is the tan delta. So the ratio between the viscous part and the elastic part of the response, which changes with frequencies. And it changes all over the place when I change the density in a relatively narrow range of concentration and the gelation rates. However, I realized that actually this type of spectra description are not completely different from each other, because in fact, I can scale the x-axis, right, the frequencies, in a different way. So with the different factors change when I consider the different gels, and in fact construct a master curve, which is what you see in the top plot. And this master curve starts to be very interesting. First of all, because I can think, okay, there is some unique type of constitutive behavior that these gels are following, and so I can think of, so I can fit my master curve and think, okay, which combination of linear viscoelastic elements, all this is in the linear response regime, would allow me to reproduce this type of master curve. And so the first thing I can think of, well, this is an elastic solid in the end, so I'm going to consider a spring and dashpot, a Calvin-Voigt model, and this will give me this dotted line here. So clearly there is some part of that, but it does not describe all the gels. And then I start to say, okay, let me add more of this. Maybe there are some time scale and then, that I'm not considering. So I add more, I add more, I add more. I realize that I need to add 1,500 of them in order to describe this type of behavior. But what is even more interesting is that, in fact, I cannot just add them randomly. I have to organize them, add them in a very organized, specific way, which is hierarchically. So the way this type of curve actually corresponds to a very specific rheological model, which is what is called a ladder model, in which these linear viscoelastic elements are organized hierarchically. So this clearly tells me that in spite of the disorder, in spite of these gels seemingly going all over the place, there is a hierarchical organization in terms of viscoelastic element embedded in this structure, which is the same across all of my gels. So these type of, and in fact, I can keep this rescaling of my x-axis and then rescale the y-axis for G prime and G double prime to confirm that this type of behavior actually corresponds to the whole viscoelastic spectrum of the system. If I take the continuum limit of this infinite amount of hierarchically assembled linear element, I obtain this type of fractional, a more compact representation of my rheological model, which is what is called the fractional rheological model. Okay, this type of ladder-like rheology has been seen in other type of gels, in particular when they are very close to the polymer networks, close to the percolation threshold, where in fact the chemical architecture is supposed to be fractal. So the idea is that this comes from a fractal type of structure in the gels. But in our gels, we can do a fractal analysis, and I will show in a minute, and they actually are not fractal. So the structure itself, if I just look at the structure and I use the whole structure to understand this type of rheological behavior, I have a hard time understanding where this comes from. And so I have to look at the microscopic dynamics, the spatiotemporal microscopic dynamics, to get a clue of where this is coming from. In fact, what we had studied with this type of simulation is the spatiotemporal distribution of the local motion of the particles. And we have realized that there is a correspondence between the fact that you have these large-scale heterogeneities, which are related to having regions where the gel is more tightly connected, so where stresses can accumulate and where the motion of the different parts of the gel is more constrained, to regions that are floppier, looser, and where things can move more. So we've spent quite some years studying this type of network-related dynamics. And we have seen that this type of characteristics, in fact, controls a lot the non-affine motion in the gels and even the nonlinear rheology. So the idea was that actually we can try to introduce that notion here by thinking in terms of mesh sizes along the network. So we see it on one node, which is something that it's possible to do in this simple model, but not always possible to do in a very, in a more complex real gel structure. So we sit on a particle and on a node of our network, and we walk along the network to measure the distances along the network between nodes. So this is kind of a topological length. And if you do these measurements across the network, you have like a huge amount of possible path going from one node to the other. We take the shortest one, and this shortest one, they already have a distribution that is quite wide, and it becomes wider as the gels actually become more and more dilute, as you may imagine, but also in general as they start to approach the rigidity threshold. So there is a situation in which is when the gel first start to become rigid. And at this point, you have the largest possible, the widest possible distribution of these length scales. As your system goes beyond the rigidity threshold, then you get more and more regions that are more tightly connected, and this makes this distribution narrower and narrower. So we take this distribution, we take the variance actually this distribution, and from this we construct a length scale, which we assumes actually control the size of these fractal objects that are embedded in the network structure and that control the rheology. So I just want to quickly show you how we gather evidence that something like that does exist in our network. So this is basically the distribution of the fluctuations of the microscopic displacement during due to the microscopic dynamics, for example, the spontaneous relaxation dynamics. And then if you change the volume fraction of the gel, or if you change the gelation rate, you get distribution that change all over the place. But as you use this length scale, so this variance of this distribution of these mesh sizes which is sort of gives you a weighted average of the size of floppy regions in your network, then you get basically all this distribution. The whole dynamics is recapitulated by a unique distribution. So if now you say, okay, this is the length scale of my actually fractal hierarchical object, and this object, become of the order of the sample size when I'm close to the gelation threshold and the system is becoming rigid, but becomes smaller and smaller as the system goes away and from the rigidity threshold. So the presence of this fractal object is some kind of memory of how the system became rigid. And so, and then it means that we can see actually our network, like if I did some different type of X-ray into my structure, I would see that it's actually composed of this fractal element, and then this fractal element gives me the rheology. So we put together a series of tests to actually verify this hypothesis and to extract the information about this fractal element. So one thing is that, for example, if I take this length scale, and if I plot it as a function of the amount of branching points that in this model corresponds to the distance, or it's a good proxy for the distance from the rigidity threshold, then I can see that this length scale psi, if I plot it just a function of the gelation rate or of the amount of particle, changes in different ways. But when I plot this for all the network just as a function of the amount of branching point, then I can see that this follows a unique scaling behavior, and it has a power law dependence that is very similar in what you would see in percolation, in random percolation, but this is a rigidity percolation type of phenomenon. And then we can do some other type of fractal analysis to extract the fractal dimensionality of these elements, and then test that against how, for example, the amount of branching point in our system depend on the amount of particle, and what we can compute in the simulation matches precisely the prediction that we have from this theoretical hypothesis. And so why is that important? Well, when you have something like a gel and a structure that is becoming rigid through a percolation process, then there is, people have studied this type of phenomena a lot, and thought that this percolating cluster that you form, in fact, can be recapitulated in this type of blob links and nodes model, where you have blobs whose size, in fact, increases as you approach the percolation threshold. And in the limit close to the percolation threshold, you have just one blob that actually is of the order of the system size. So this is where also our length scale becomes of the order of the system size, because there is just one object that is responsible for the rigidity of the system. If I cut that object, that's when my system becomes not rigid. And as I go away from the rigidity threshold, I get more of these objects that are smaller. So the way percolation people characterize that is in terms of the amount of these singly connected bonds. Basically, in these type of structures, not all the bonds are equivalent. There is hierarchy of bonds, and in particular, there are bonds that if I cut them, they interrupt the connectivity. In this case, they interrupt the rigidity. And they found out that these bonds are actually hierarchically organized. And they are organized hierarchically over length scale that become larger and larger as I approach the rigidity threshold. So using this, so the idea is that these structures are important for the mechanics of the system, because basically they tell us about where and how these singly connected bonds are distributed. And the presence of these singly connected bonds tells us about the floppy modes in our system. And so in fact, we have done a little calculation. Bhavan did a little calculation of the floppy modes of our imagine this elastically active backbone that is in each of these blobs, and how does that scale with the blob size. And what you find is that the soft modes associated to that are again hierarchically organized, and they are hierarchically organized, interestingly, precisely in the same way as our rheologic, viscoelastic element in the ladder model. So why this is useful? Well, it's useful because now I can say, okay, maybe to predict the rheology of my system, I don't need necessarily to know where each particle is, but I need to have this information that the structure that I see contains these hierarchically organized elements, which gives rise to that type of viscoelastic response. And so if I imagine that my network is my gel is a percolating network of these blobs, I can use basically, and in our case, there are these bending interactions that are associated to the bond stiffness. So I can use one of the models that people have developed for percolating network to compute how the modulus should scale as a function from the percolation threshold, in our case, the rigidity threshold and the blob size. And so I have a way to predict now this exponent with which the modulus should depend on the distance from this rigidity threshold. And if I look at my shift factors in my master curve, the y shift factor is basically the modulus of my gel, which scales precisely in this way as a function of the amount of branching point or as a function of the size of the blobs. Therefore, explaining that that's precisely where the rheological response comes from. What is very interesting is that I also have an horizontal shift factor, which is a time scale. So there is a hierarchy of time scale because there is a hierarchy of these viscoelastic elements. And this hierarchy of time scale, this time scale is actually, we can verify in the microscopic dynamics that it's associated to the viscoelastic delay time. So how long does it take to the system to realize that it's actually elastic? So how long things can flow because they are poorly connected. And this seems to scale in the same way with the same exponent of the modulus, which is something that I would like to understand more. I don't think it's ever been tested in experiment. I imagine that one could measure this type of time scale in microbiology experiment, and I would be very interested. The other thing that is very interesting in this type of result is that now you think, okay, you have obtained this scaling, but it's mainly because we were able in the simulation to identify the rigidity threshold and have a parameter that measure the distance from the rigidity threshold. But how do I know that, especially if this depends on not necessarily on all the structure that I see? So the idea is that in the experiment, what I typically will measure is the modulus as a function of the amount of particles, right? This is what I can, or the concentration. This is the parameter that I can control experimentally. And so the idea is that when I see power laws in that, it's some kind of apparent power laws that depends then on how the structure that I see has been formed starting from this backbone that determines the rigidity. And so, for example, that's an interesting hypothesis because then this means that these two scaling should be related with each other by this type of scaling relationship in the case in which the structure that I'm forming is, for example, happening through a fractal aggregation process, which would mean that then the exponent that I measure in the experiment and the dependence of the modulus on the amount of particles, it's not necessarily what I predict here through this model, but it's a combination of this exponent with the fractal dimension of the things that that the system start aggregating from. And so these are examples of what are the values that, for example, I would have if I have my colloidal system that it's aggregating through the LCA or RLCA type of processes. And then I would get an apparent exponent of how the modulus depends on the volume fraction that can vary enormously, right? And this is very much consistent with what is seen in experiment on colloidal systems, where in certain cases you see exponents that are actually already very close to this 3.5 that is predicted by this percolation theory. And other cases, they're very far from that and they're much larger. And so this type of explanation that depending on how your aggregation takes place, you are going to see different type of dependence of the modulus on the amount of particles that are there would allow you to actually capture this complexity. So it's always a type of rigidity transition and this type of rigidity transition, but you are going to see a different exponent because the aggregation that builds the rigid structure is different.
You have about, oh, you already had the summary slide.
Yes, yes.
You're good.
So I just wanted to summarize with just a thought, and this is something that we wrote for the blog for the Nature Physics paper that went with this result that I just told you about. So this idea is that you can look at your gels and there are actually some, and think that there are some hidden fractal structures in that actually predict its mechanical response. And it's a bit like saying that there is some kind of cosmic wave background in there, and that this type of analysis will allow you to unravel that, but not only, but that you need to have access to that to actually predict the response of the gel. I want just to conclude by actually thanking my collaborators of things that I've mentioned that I've not mentioned. And in fact, just to, advertise things that we're doing currently in my group, which have to do a lot with the composite network. And this is especially thanks to Mauro Mugnai, that is a fantastic computational physicist we started recently to collaborate with, and gels with much more complex adhesive interactions that Vinuta has been pioneering in my group. Thank you a lot.
All right. Thanks, Emmanuel, for the wonderful talk. I think we have two questions in the chat for your research talk. I think both of them are towards some of the earlier slides. The first question is from Norris. This question is, are these regions of tension accumulation related to local particle density? I believe this is about an earlier slide where you talk about tension buildup.
Yeah. So maybe I can use that to show some of the things that I wanted to show in the tutorial, but I was going too slow. But I think it's, let me just stop sharing. Mm, 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 mm. So basically, I hope you can see this movie. Let's see. Right. Okay, so basically this is our gel, and this is colored in terms of the local amount of branching points. So you see regions where you have a lot, the red regions where you have comparatively much more branching points, and regions which are blue where there is very fewer branching points. And so this is correlated to density, but it's not just density of particles, it's more density of connections, right? And it's a density over larger scale, right? Because even if I have a, like regions where like a particle that has few more branching points, but then it's connected to a region that it's very floppy, that's not going to do much, right? And so, for example, from here, we did the statistical analysis of what's the tendency of different parts of the gel to break. And you get this type of, now thicker is where it's more likely to break. So you can see that statistically, the regions where it's more likely to break, they tend to be yellowish red, right? So it tends to happen more in regions that are more connected, which is kind of counterintuitive a little bit in the beginning, but it makes sense if you measure tension and you can see that locally the network is pulling in different direction because there is more connections there. And that's why basically higher order connections there. And that's why you get basically this type of effect. So that's where the tension tends to build up. But I want to emphasize that this is not, and that's what made this analysis very hard. And now it all makes sense in the things that we're doing. It's really depend on the network topology and not the local coordination. So just to look at what one particle does locally or even on average over the network, it's not enough. You really need to identify larger scale motifs that form the topology of the network in order to understand how this works out.
Am I understanding correctly that these highly connected regions are more prone to break because they're more solid-like, and the sort of bluish region, because you have less connection, so it's more like a liquid-like, and so
Yeah,
if you add tension, it's just going to be somehow dissipated, like
right.
in
Yeah,
sliding, right?
absolutely. So let me show you this, that then when you break things in this part that are like yellow and red, you could do another analysis to say, okay, where do things move mainly, right? When things break, where do I see most of the rearrangements? And then when you do that, you get this blue sausages. So telling me that you break things where they're red, but then the motion propagates away from that in the regions that are floppier, because that's where I can accommodate more rearrangement. And because of the network connections, I can actually go very far away from that. So that's how basically the idea that you can get hierarchies or timescales and length scale in this system then becomes more visual. And it's very interesting for me to think that then these materials are not just about organizing where particles are, but they are about organizing timescales and length scale, right? And if you think about how these materials are present in the biological context, that's kind of very intriguing, right? Because they contain a lot of information that actually can be used, for example, by cells to time things or to integrate things and so on.
And I would just like to comment, like in the biological gels, you also have like active motors, things that's adding more time scales.
Exactly. And it's, you can think that this is what I really want to do, to put active processes in these networks. And you can really, think how, in fact, those active processes, yes, can break things or recombine things locally, but how that is going to change the larger scale or organization and the topological motifs in the network. I think this is something that it's very interesting to me.